Zo, dames en heren, gaat u zitten, als u be seated, please. Dames en heren, geachte collega's, ik heet u van harte welkom op de openbare doctoraatsverdediging van Gabriele Nasello en verklaar de zitting voor geopend. De heer Gabriele Nasello, master in biomedical engineering van de Politecnico di Milano in Italië, heeft aan de KU Leuven en aan de Universiteit van Zaragoza in Spanje een proefschrift voorgelegd handelend over bone regeneration in patient-specific scaffolds, from microfluidics to computational simulations. In het Nederlands heet dat botregeneratie in patiëntspecifieke scaffolds, van microfluidica tot computersimulatie. En dit tot het verkrijgen van de graad van dokter in de ingenieurswetenschappen aan de KU Leuven en dokter in biomedical engineering aan de Universiteit van Zaragoza. Hij zal dit proefschrift nu verdedigen. I'm asking on the candidate to first give a presentation of about 45 minutes about your work, after which there will be time for questions and discussions. So, Mr. Gabriele Nasello, you have the floor.
Yeah. Thank you. Okay. Oh, you can hear me, I guess. Perfect. So thank you. And yeah, dear chairman, dear jury members, dear colleagues, friends, family, welcome to my PhD public defense. So first of all, I would like to thank you all for attending this event, both physically and virtually. And now I'm going to guide you through all chapters of my PhD thesis. And I will show you how I use multiple experimental modeling techniques to study the bone regeneration process for patient-specific orthopedic applications. So based on the chapters of my PhD thesis, I divided this presentation in 4 sections. I will start with a brief introduction about the topic of my PhD, and it will cover chapters 1, chapter 2. And then later, we will move to the chapters where I describe the research that I carried out. So we'll move to chapter 3, chapter 4, and the 2 types of experimental models that I used. Finally, we will move to the final chapter, chapter 5, where I draw main conclusions based on the results I present to you, and then I will provide some recommendations for future works. So, as bone is the first word of my PhD title, I would like to start with this presentation with 3 key concepts about bone tissue. First concept is about bone structure. So bone has a hierarchical structure growing from the macro to the nanoscale. But for the sake of this presentation, we just need these 2 types of bone structures at the macro scale. We need trabecular bone, which is a less dense type of bone, and then we have also more dense, compact or cortical bone. As I said, these are just macro scale different types of bone structures. And you can imagine that the 2 different structures, they have different structural function, mechanical function, so they will be distributed in a different position throughout the skeleton and even in each bone. Second key concepts regard the bone cells that are responsible of resorbing the bone matrix, and those are the osteoclasts, forming the bone matrix, and those are the osteoblasts, and then regulating also this formation and resorption process, and those cells are the oocytes. So later in this presentation, I will focus more on these last 2 cell types, on the osteoblasts and the oocytes. The third key concept regards bone diseases. And here you see 2 examples, osteoporosis and bone fractures. In general, we can say that during those bone diseases, the regulation of the bone cell types on the bone structures, and therefore also the bone structure, they are highly altered. So bone diseases are probably one of the major causes of pain and disability in Western countries. And unfortunately, in extreme cases of pain, the only solution available is, of course, surgical. Patients need a surgery. And in those cases, especially to restore a joint functionality, we do need orthopedic implants. And those ones I'm showing you here in the slides are probably the gold standard of orthopedic implants. Those are 3D printed orthopedic implants. And the great advantage of those devices, of those implants, is that they can match complex macro-scale geometries. So they're extremely successful in treating patients that need a personalized treatment. But another great advantage of those devices is that they have, or we can fabricate, thanks to 3D printing or additive manufacturing, that's the same, we can fabricate a porous structure at the bone implant interface that enhances and facilitates the osseointegration of those implants. So research is now trying to apply the principles of tissue engineering to 3D printed orthopedic implants to maximize this osseointegration. But what's tissue engineering? So for this presentation, I want you to know about the concept of in situ tissue engineering. The rationale behind in situ tissue engineering is that a graft is implanted in a living organism, and this graft can be whatever. It can be a biomaterial, in that case we call it scaffold, but it can also be something developed by cells. Everything implanted in a living organism that induce endogenous cells, so cells from the host, to invade the graft, that's something we use in an in situ tissue engineering approach. So the graft has only to recruit cells from the host that will invade the site where the graft has been implanted, but the cells from the host, they will regenerate and create new functional tissue. So you can imagine that the final regenerative performances of those grafts, they will depend, of course, on the graft properties, but also on the patient's characteristics, because the patient cells, those will be the ones that will form and heal the tissue. Of course, if we talk about 3D printed orthopedic implants, we will call them scaffolds because we will use the biomaterial-based approach. So the final regenerative outcome, it will depend, of course, on the composition, so on the biomaterial composition, on the microstructure we will give thanks to 3D printing, on the mechanical properties, but it will also depend, probably even more, on the characteristics of the patients. So host cells, they will form new tissue, and of course, depending on the age of the patient, on the presence of comorbidities, the final outcome can be totally different. So we need to study and to model both scaffold and patient. So in tissue engineering, we can identify 3 different categories and types of experimental models, starting from the in vivo models, those are in animals, and of course, they're probably the closer, or they represent a complete biological environment. The great disadvantage of this model is that, besides ethical concerns, that sometimes this complete biological environment or the biological mechanisms that are taking place in an animal are not always the same ones as the ones occurring in humans. So we can also test human cells. We can do that also outside a patient if we want to be sure that the biological mechanism we are studying is the one taking place in humans. In that case, we can use an in vitro model. So that's, of course, a clear advantage of in vitro model. However, you understand that we will never have all the factors that makes a complete biological environment. So we have higher control because we are culturing our cells in a laboratory outside a living body. But of course, not all factors will be present. And then after, we have in silico models. In silico models, they allow us to isolate a single mechanism and to study its contribution on a global and more complex biological process. But of course, those models, they have disadvantages, such as the cost of development, because developing those models and then also assess and validate those models, so quantify their credibility, this process takes a lot of time. But we will see a little bit more of this later in the presentation, on how these models are built. So after this introduction about bone tissue, 3D printed orthopedic implants, and the rationale of applying in situ tissue engineering approach there, and different models of scaffold and the different types of models, I can now state the global objective of my thesis, which is the evaluation of the bone regeneration process guided by 3D printed bone scaffolds for patient-specific orthopedic applications. So as I said before, we have these 2 factors, both scaffold and patients, that will affect the final regenerative outcome. And therefore, I divided my research into testing 2 different hypotheses, and each one focusing on one of these 2 factors. So in the first research hypothesis, I tested whether the mechanical interaction between the scaffold and the host might determine the final regenerative outcome. And that was for patient-specific and even implantation-specific, so for location-specific applications. So since I wanted to isolate the mechanical problem of a more complex biological process like bone regeneration, I used an in silico model to do that, which was later calibrated using in vivo data. For the second research hypothesis, I tested whether we could culture human cells, and we could use human cells, so primary human osteoblasts, isolated from single donors, to study and to quantify the osteogenic activity, so the patient-specific osteogenic activity. So I wanted to use human cells outside, of course, a patient, and that is why I used an in vitro modeling technique. And more specifically, I used a bone-on-chip, an advanced microengineered system that I will describe a little bit more later in this presentation. So as you can see, throughout my thesis, I used different experimental modeling techniques. And now I will give you a brief overview of the research that I carried out, and I will also describe the technology I used. So let's start with chapter 3, where I performed my study with the in silico model. But before starting, let me just mention that the research I'm going to present to you now has been already published in a research manuscript, where I'm the first author. So, as I said before, the idea of applying additive manufacturing or 3D printing to bone tissue engineering has already been quite developed. But the initial idea was to fabricate scaffolds that were matching the mechanical properties of the replaced bone tissue. But you can see here that it's pretty easy to do. Of course, with 3D printing, we can change the fabrication parameters, the microstructure, in order to match these mechanical properties. But when these scaffolds were implanted in vivo, you can see here the outcome on the right side of the slide. The scaffold matching the mechanical properties of the replaced bone tissue is the one with stiffness of 1400 megapascal. That wasn't the scaffold with the highest regenerative performances. But softer scaffolds, they were performing better. The second interesting result that I will see from this study is that authors could identify two different types of responders. So they divided all the animals they tested in two different groups. There was a first group of weak responders, where the final regenerative response didn't depend on any of the mechanical stiffness of the scaffold. So for all values of mechanical stiffness, the final outcome was always the same. There was another group, which was called strong responders, where the final response depended on the scaffold stiffness. So the lower the stiffness, the higher the response there. So if you already have such variability in the response in a study with just 12 sheep, it really means that the host makes a huge impact in the final response. So we need really to isolate the mechanical problem and study the contribution of the patient. You can see here then how we build an in silico model, because that's probably the ideal technology to do that. So building an in silico model consists of several steps. We can start from creating a model, and the phase of model creation starts, of course, from a biological question, a biological process that needs to be translated into mathematical equations. And once we do that, we usually have model parameters of the mathematical equations. And then when we solve our mathematical equations with a computer, we then need to compare the models of the parameters. So we need to compare the numerical output to experimental data. In this way, we do a calibration. We can find the values of these model parameters. After that, we can run again our analysis. We can compare with other experimental data. And what we do at the end is a validation. And once we validate our model, we can state that our in silico model can predict the biological process we are studying. So let's see how I apply this whole process to my research. So the idea was to study the contribution of mechanics in bone regeneration driven by scaffolds. But we wanted to check the effect of both the host and the scaffold. So the first two assumptions I did to build my model, to make a translation to mathematical equations, were that first, the local mechanical stimulus was dictating alone the distribution of the bone ingrowth inside a porous scaffold. That's a big statement. But of course, if we want to isolate a mechanical problem, that's what we have to do. And the second assumption was that if we want to model the patient-specific response, we need to use patient-specific parameters. And then, of course, we need to compare our simulations, our in silico model, with experimental data, and that's why I used the data coming from an in vivo study performed by Materialize, where 3D printed porous titanium scaffolds were implanted in the distal femur and proximal tibia of 3 different goats. So let's see how we built this model, how we built these mathematical equations. So we have 3 components in our model. The first one is the bone tissue, then we have the 3D printed porous scaffold, and then we filled the pores of this 3D printed porous structure. We filled the pores with a granulation tissue. And this is the third component. So this granulation tissue, immediately after implantation, has very low mechanical properties. And then over time, as the tissue gets mineralized, you will see that the mechanical properties of this granulation tissue will start increasing. So the equation driving the mineralization process of the granulation tissue consisted of 2 components. We had a biological component, which was made by bone-forming cells invading the granulation domain. So we were assuming that immediately after implantation, no cells were present inside the scaffold, and then over time, cells were diffusing. And that's why I used a pure diffusion equation to describe this biological component of the model. For the mechanical component, I adapted the mathematical formulation of the bone remodeling theory to bone regeneration. So the bone remodeling is a physiological process that takes place throughout your body. And what happens usually is that in a fully developed bone matrix, there are processes of bone resorption and bone formations that take place. And so a mathematical formulation of a purely mechano-based law assumes that you have a reference stimulus named psi star. So if the mechanical stimulus in your area is higher than this reference value, you will have net bone formation. While if your mechanical stimulus is lower than this reference value, you will have net bone resorption. So when translating this mathematical formulation to bone regeneration, first I assumed that no bone resorption was taking place during the regeneration process. And that's because I was starting with granulation tissue, and I was assuming that no resorption was taking place there. So you can see here in the graph only the part where tissue gets mineralized when the mechanical stimulus is higher than a reference value named psi star. But then I cannot assume that the mechanical stimulus initiating bone formation in bone regeneration can really be the same of the one initiating bone formation in a fully developed matrix, as it happens in bone remodeling. So I reduced these values by a factor, alpha, which is the first parameter of the model, and I assumed that this parameter could vary depending on the implantation site. So that's a first parameter. We have a parameter that can change depending on the location where the scaffold is implanted. Second parameter of the model is the constant that relates is mechanical stimulus and tissue formation and mineralization. So this indicates basically the sensitivity to mechanical stimulation and make it a patient-specific parameter. Because I was telling you that the host will have a tremendous effect on the final response. So I assume that my 2 model parameters in this case, so the sensitivity to mechanical stimulation and the reduction of the stimulus initiating bone formation, to be patient-specific and location-specific parameters. So after looking at the mathematical formulation, we can finally build the model. And that's when we have to apply boundary conditions to our domain, which are conditions at the extremities of our model. And we need to apply these both for the biological part of the model and for the mechanical. So for the biological, you can see here on the top left, and you have the granulation domain, and I was setting that immediately after implantation, the concentration at the bone, at the interface between granulation and bone tissue was maximum, because cells there were present, but so the concentration was 0 throughout the granulation domain. So cells over time could invade inside, those were bone-forming cells, and then where the mechanical stimulus was quite high, then tissue mineralization was taking place. The other boundary conditions, they regard mechanical loads, and you can see here where I applied loads, both in the femur and in the tibia of the goats I was simulating. And those loads were proportional to the body weight of each animal. But before continuing, I would like you to notice that the locations where those scaffolds were inserted in the in vivo study. You can see the scaffolds were inserted both in the femur and in the tibia, and we can identify two different implantation sites. Scaffolds like the ones in the blue and in the green location, they are surrounded by bone with pretty uniform mineral density and very low mineral density. It's quite typical of trabecular bone, and that's why I call this location epiphyseal. The other location, like the orange, you can see there that the mineral density isn't that uniform. It's more heterogeneous, it's higher in the outer side of the bone. We're getting close to the diaphysis, we're getting close to cortical bone. And that's why I call this location more diaphyseal. And you can imagine that the mechanical states in these two locations would be quite different. So now we can finally start our simulation. We can go on with our analysis. Of course, I simulated 12 weeks, like the duration of the in vivo study. And then at the end of these simulations, I need to post-process the data in the same way as the in vivo study has been done. So I created a so-called in silico micro-CT. So I analyzed the data in the same way as the in vivo study has been analyzed. I could then separate the mineralized part from the not mineralized part in the granulation domain. And then I could quantify the bone ingrowth in 3 different subregions, in the medullary, middle, and periosseal subregion, where the periosseal is the region closer to the outer surface of the bone. So we can now get finally the results of our model. You see, it's a very long process that takes time also to be explained. And looking at the results, you can see that this mechano-driven model of bone regeneration, it predicts a uniform bone ingrowth distribution when scaffolds were inserted in the blue and in the green locations in the epiphyseal location. While there is a clear shift in the bone ingrowth distribution, which is localized in the periosseal region when scaffolds are inserted in the orange location, in the more diaphyseal location. But what I want you to notice is that we have parameters in our model. So when we change those parameters, the ones I mentioned you before, when we change those parameters, of course, the outcome will change. But this trend of uniform bone ingrowth in the epiphyseal location and localized bone ingrowth in the diaphyseal location, this is consistent for basically all the values that I tested for the model parameters. So it really means that the mechanics is behind this problem, is really probably describing what's going on. Another key result of this model was the interpretation that I provided for the limited bone formation inside the core of the scaffold. So this has been observed both in my simulations and in vivo, that no bone was formed in the scaffold core. And if you can see here in the dashed lines, this is the numerical output. So this is the value of the bone ingrowth at each specific iteration. Of course, this can be done in vivo. We can't check the in vivo ingrowth over time. But you can see that for my model, most of the bone ingrowth is taking place in the first 4 weeks after implantation. While in the points here in the plots, they represent the final in vivo outcome measured in animals. And this model would explain why no difference in the bone ingrowth has been observed after 6 and 12 weeks from implantation. But then, as I told you, we need to compare our model to experimental data. And when we do that, we can find values for our model parameters. So we are doing a calibration. And when I calibrated my model, you can see here in the bottom the value of the parameter that represents the mechanical sensitivity of the host. You can see here that one animal showed higher sensitivity to mechanical stimulation. And of course, that animal was the one reporting also the highest values of bone regeneration, of bone ingrowth in the scaffold. Regarding the other parameter, the parameter that was reducing the mechanical stimulus initiating bone formation, if you can see here in the slides, the mechanical stimulus, it looks like the mechanical stimulus initiating bone formation in the femur was consistently higher in all animals than the mechanical stimulus initiating bone formation in the tibia. So it really looks like that we want to test, we want to predict the regenerative performances of a 3D printed put of scaffolds. We need to know first how sensitive is the host to mechanical stimulation, and second, where this scaffold is going to be implanted. So just to summarize a little bit the main findings related to this chapter, Well, this mechano-driven model of bone regeneration, it does predict differences in the bone ingrowth distributions based on the implantation site. It provides an interpretation why there was limited bone ingrowth in the scaffold core. And then it could identify one animal with higher sensitivity to mechanical stimulation compared to the others. So of course, if we want to use this model then to optimize the design of a scaffold, we need, as an input of the model, information regarding the patient, information regarding the sensitivity of the host. And this motivated the second research of this thesis. So the following chapter, we can now move to chapter 4. And again, before starting, I'd like to say that the results I'm going to present to you now has been already included in a research manuscript where I'm the first author. So, if you recall, before I was saying that in our in silico model, we were just modeling bone formation. But what is bone formation? Like, what are the cells responsible for forming new bone tissue? Those cells, they all arise from skeletal progenitor cells. And once they are committed to form new bone, they first differentiate into osteoblasts, which are the bone-forming cells, the ones that actually mineralized, and they form newly mineralized bone matrix. And once osteoblasts are fully embedded in their own, or surrounded in their own mineralized matrix, they will finally differentiate into osteocytes. So you have those cells that from skeletal progenitors, they differentiate into osteocytes, and then osteocytes is the final differentiation stage. And of course, this differentiation is characterized by stages and changes in the cellular phenotype. And here you see some examples of those changes. They regard cellular morphology, cellular proliferation, and protein synthesis, which of course comes from a change in the gene expression. Those changes have already been widely studied in vitro. Nowadays, there is a recent advanced in vitro modeling technique named organ-on-chip, where micro-engineered devices are combined and are used to culture cells. So in those devices, we can create complex culture microenvironments where we can replicate specific aspects of organ function. Of course, we don't want to replicate the whole organ, but we want to just replicate a specific aspect. And to show you an example of this organ-on-chip, of a bone-on-chip, in this system, it was possible to co-culture at the same time different cell types, osteocytes and osteoclasts. They were cultured in separated channels that were communicating with each other, but moreover, it was possible also to apply steady fluid flow just to a specific cell type. So at the same time in this system, the authors could culture mechanically stimulated osteocytes, osteoclasts, and not mechanically stimulated osteocytes. So in this system, you have a high control of the culture environment where you can apply complex mechanical, chemical stimuli. So my idea was then to culture cells isolated from single donors, primary human osteoblasts, and use a bone-on-chip to assess the osteogenic activity of that specific donor. And the use of the organ-on-chip, the bone-on-chip, was critical because those devices, they required a very limited amount of cells. So it makes it quite easy from a small biopsy to get cells, to get osteoblasts, and then culture them in many devices. So, of course, I wanted to capture variations between cells isolated from different donors. But, of course, I wanted my model, my bone-on-chip, to replicate findings that are already quite accepted in the literature regarding, of course, culturing primary human osteoblasts in vitro. And one of those findings is that the cell seeding density affects osteoblast differentiation into osteocytes. So, of course, I wanted my model to do the same. But let's see how we build, how we fabricate a bone-on-chip. The technology, the microfabrication technique used is pretty standard. It's called soft lithography, and it starts from a master where a microengineered pattern is etched. And then in this master, we can pour a soft elastomer named polydimethyl methylsiloxane, or PDMS. And after curing this PDMS, what we have a solid polymer, a solid soft elastomer, that is incorporating this micro-engineered pattern. So once we punch each single device and we create the inlets where we can insert our cells or the media, we can autoclave those devices and then bind the device, thanks to plasma surface treatment, to a glass dish. So at the end, that's the device that we have. It's quite small. And the culture environment is basically confined by the glass dish on the bottom and then from the micro-engineered pattern that is incorporated in the PDMS. So let's see a little bit more into the details, the components, the different culture chambers, the different chambers of the bone-on-chip. We can identify in pink the reservoirs where I was placing the cell culture media. And of course, since I was inducing osteogenic differentiation, I was using osteogenic media. And there, in purple, you have the area where I was introducing my primary human osteoblasts. But given that in these devices we can also load hydrogels, my osteoblasts were embedded, were encapsulated in a collagen type 1 matrix, in a collagen type 1 hydrogel, before encapsulating in the culture chamber. So at the end, once I was introducing my cells, I had in this small device, in small chamber, I had primary human osteoblasts cultured in 3D in a collagen matrix. So as I told you before, I wanted to study and to test whether the cell seeding density was affecting osteoblast differentiation, and that's why I seeded my cells at 2 different concentrations, taken, of course, from the literature. I seeded cells at 1 million cells per ml and at 250,000 cells per ml. And I wanted also to capture and to see if there were variations for cells isolated from different donors. And that's why I repeated my experiment with cells isolated from 2 different donors, a female donor, 86 years old, and a male donor, 73 years old. And then to assess this osteogenic differentiation, I evaluated some of the characteristic changes that I listed before. And of course, I started from the cellular morphology, because it's probably the most evident change that you can observe in your samples. So after 21 days of culture in a bone-on-chip in osteogenic media, those osteoblasts cultured a higher cell seeding density, so at 1 million cells per ml, they were showing a characteristic dendritic morphology of the osteocytes. And this wasn't happening for both donors for cells cultured at lower cell seeding density. But of course, I wanted to prove a consistent change in cellular morphology. And that's why I developed an image analysis software that could track, could compute the primary protrusion length of osteoblast from a bright field images. And you can see here that after the user was selecting manually the contour of the cells, the software was able to extract the primary protrusion length. So over time, you can clearly see that only cells cultured at a higher cell seeding density, they were significantly increasing the primary protrusion length over time. And the overall increase for cells coming from both donors was about 1 micrometer per day, while the increase in primary protrusion length wasn't statistically significant when they were cultured at lower cell seeding density. I also would like you to notice that cells isolated from the second donor appears to increase the primary protrusion length faster than cells isolated from the first donor. But we will see this even later. Of course, cellular morphology isn't enough to state that our cells are differentiating faster or slower to osteocytes. We need to check also some of other characteristic changes I listed before. And the other one I checked was cellular proliferation. In general, when cells are differentiated or are differentiating, they interrupt proliferating. So the osteocytes are the final stage of differentiation, and usually when cells are osteocytes, they don't proliferate anymore. And you can see here that by measuring the DNA content, I noticed that cells cultured a higher cell seeding density, they weren't increasing their DNA content over time, while osteoblasts cultured a lower cell seeding density and isolated from both donors, they showed an increase in the DNA content at the end of culture. So yes, also here, those results might suggest that our osteoblasts culture the higher cell seeding density, they're differentiating faster. But again, morphology, proliferation, it's not enough, nothing related to bone. We need to start looking at bone markers. And one characteristic marker that is usually assessed is alkaline phosphatase activity. And you can see here, so alkaline phosphatase is a protein synthesized when osteoblasts need to mineralize the matrix, and is a clear marker of osteogenic activity and then differentiation. So the alkaline phosphatase activity was higher for osteoblasts cultured at higher cell seeding density for both donors. But I want you to notice that cells isolated from the second donor, they showed higher alkaline phosphatase activity at all time points and at both conditions of densities compared to the first donor. So it really looks like that there is a difference in the alkaline phosphatase activity. Of course, this can be assessed even in a traditional in vitro system. That would be also possible. But here, we only need 10,000 cells per sample to make this to quantify this. Now, after that, we move to fluorescent staining. So 4 days before each time point, so day 7, day 14, and day 21, I added calcine to my samples, and I wanted to show, and you can see here the white dots in the images, that they show that those osteoblasts have mineralizing activity throughout the culture. Moreover, then I also stained for a specific protein that is a marker for osteoblasts, and this is the bone sialoprotein 2. And you can see here that this protein was expressed by osteoblasts cultured at lower cell seeding density throughout the culture. While when I looked for an osteocytic marker, when I looked for the dental matrix protein 1, or DMP1, that was expressed only by cells cultured at the highest cell seeding density. Here I'm showing just the results from one donor, but I mean, you can check in the thesis. It's the same also for the other donors, for the other donor. So only really if we take together all the results that I mentioned here, so change in cellular morphology, protrusion length, proliferation, alkaline phosphatase activity, and then the synthesis of osteoblast or osteocyte marker, then we can state that for the first time, I showed it in this system, in this microengineered device, we can culture primary human osteoblasts in a 3D matrix and induce their differentiation into osteocytes. As in traditional in vitro system, the differentiation depended on the seeding density. So the higher the seeding density, the faster the differentiation. And the idea behind all this project is that we can really use it to culture cells isolated from specific patients. We need just 10,000 cells per sample. It's a very limited amount. And we can use it to capture variations in the osteogenic activity, so in the osteogenic potential of different donors. So after this discussion on the research that I carried out, we can now move to the final section of the presentation in chapter 5, where I draw main conclusions and I provide some recommendations for future works. Okay, just to recap, I said that that both scaffolds and patients are affecting the bone regeneration process. We need to model both of them, and we have different experimental modeling techniques. We have in silico models, we have in vivo models, and we have in vitro models. So in the first research chapter, in chapter 3, I just isolated the mechanical problem, and I noticed that we can use this mechano-driven model of bone regeneration to predict the regenerative outcome based on scaffold implantation and host sensitivity. And that's why then I used, as I said, in silico model and then in vivo data to calibrate the model. In the following chapter, in chapter 4, then I cultured primary human osteoblasts isolated from single donors. I cultured them in bone-on-chips, and I wanted to use this device to assess the osteogenic activity of each specific donor. Then if I bring you back to the global objective of the thesis, studying the bone regeneration process driven by 3D printed scaffolds, well, we cannot leave out of consideration the role of the patient in the variability of the regenerative response. And that's why really in my thesis, I try to use different modeling techniques to study this process from different perspectives. And here I conclude that the integration of those models might be essential to maximize this mechanobiological optimization of 3D printed porous bone scaffolds. And now I want to give you an example of how those two models can be first developed and then integrated more. So regarding the in silico model, a clear future work that should be done is to test the regenerative performances of different scaffold designs for different implantation sites. Because as I presented already, the implantation site has an effect in terms of local biomechanical demand. So I would expect that the same scaffold implanted in different locations will perform differently. So probably the optimal scaffold, we will have different optimal scaffolds for different implantation sites. Regarding the other project I presented, the bone-on-chip, the natural consequence or the natural continuation of the work I presented consists of using the microfluidic properties of those devices. So those systems, as the one I presented here, they are done to apply interstitial fluid flow in the culture chamber. So if we connect the bone-on-chip to external pumps, we can have a 3D microenvironment where primary human osteoblasts isolated from single donors are subjected to steady or oscillatory to interstitial fluid flow. So we can evaluate the osteogenic activity of those cells and the mechanical stimulation. And we can use this system to estimate the mechanosensitivity, so the sensitivity to mechanical stimulation for that specific patient. And that's probably where I would combine the two models. So let me show you a real case where probably this combination could be used. So far, 3D printed orthopedic implants, they are used in the clinics for two-stage revision surgeries. So those are surgeries where we have first to explant a bone tumor or a previous implant that failed, but we cannot implant a new orthopedic implant immediately. The patient has to wait from 2 to 6 weeks before the implantation of a new device, and during this time, a patient-specific device is produced. So usually after the first surgery or explantation, the final bone shape is quite not standard, it's very complex because you remove the tumor, because you remove the previous implant, so you took out some pieces of bone. So the patient needs a personalized treatment. During this phase, when we are designing and developing the patient-specific scaffold, we can also test. So if we take just a very small biopsy from the patient during explantation, we culture our bone cells in a bone-on-chip, we apply fluid flow, we can estimate the mechanosensitivity of the patient. This is one of the two inputs of my model. We can use it as an input for the computational model, and then we can use this information together, of course, with the anatomical site where the scaffold will be implanted. We can use this information to build a mechanobiologically optimized 3D printed orthopedic implant. So let me just conclude this presentation by summarizing the scientific output associated to this thesis. I already mentioned the two research manuscripts I published. I completely forgot telling about, although they were in the slides, I completely forgot about the 2 literature reviews where I also contributed. One was regarding the development of computer-based models for bone regeneration, and the other one was for the design and the development of hydrogel-based bone-on-chips. And before COVID, I could also attend 8 international conferences, and I was also awarded by the European Society of Biomechanics with a travel award, and I was recently nominated also for the student award. Last but not the least, let me just mention all the academic and not academic institutions that were involved in my Marie Curie European Industrial Doctorate. Of course, I would like to start with the University of Saragossa, my host institution. After that, I moved to the National University of Ireland in Galway, to Materialise Company, then to Cow Leuven, and to the Robert Jones and Agnes Hunt Orthopedic Hospital in UK. This brings me to the end of my presentation. Again, I would like to thank you all for your attention, and now I would be happy to answer your questions.